In the name of the Father, and of the Son, and of the Holy Spirit. Amen. Lord, grant us concentration, that we may learn, listen, understand, have a peaceful mind, and remember that you're with us always. Amen. Okay, so we've talked about the light reactions, the use of the light energy to energize the electrons, passing them through an electron transport chain to both reduce NADP to NADPH and to create the proton gradient needed for ATP synthesis. That was the light reactions using the two photosystems. Okay. Then we talked about Kelvin cycle, the carbon fixation through the process of Rubisco, the reduction of the intermediates that were formed using the energy in ATP and NADPH, which were formed in the light reactions, and then the regeneration of the starting material with one for each cycle, one glyceraldehyde 3-phosphate being available for glucose synthesis. Okay? And so we've already talked about the steps in photosynthesis. Now there's one consideration we need to make, and that consideration is Rubisco itself. Okay, as I mentioned in the last video, Rubisco is also known, or the full name, if you will, is ribulose bisphosphate carboxylase oxygenase. Okay, and so we've already talked about the carboxylase activity. The carboxylase activity is carbon fixation. Okay, and so that's sort of the normal process, if you will, that we want Rubisco to go through. However, there is a problem. Okay, and that problem is that Rubisco has a low affinity for carbon dioxide. Okay, what that means is for carboxylase activity to occur, there needs to be a fairly high concentration of carbon dioxide inside the cells. Okay. And the way that this is generally regulated is by the opening and closing of the stomata. The stomata are the openings on the bottoms of the leaves that allow gas to come in and out, and it's also going to cause some water to be lost. Okay, and so what happens is in hot weather they will close, which will prevent the carbon dioxide from coming into the cell. Okay, so the carbon dioxide can't get in, and the oxygen can't get out. And so inside the cell, or inside the leaf, we see a high concentration of O2, okay, and we have a low concentration of CO2. And so as long as the stomata are closed, no gas exchange is occurring, which means the oxygen is building up and the carbon dioxide is becoming low. And this is where Rubisco is going to shift. Rubisco is going to go from carboxylase activity to oxygenase activity. Whereas the carboxylase activity uses the ribulose bisphosphate to fix carbon to it, the oxygen is actually going to break down ribulose bisphosphate. And that's going to result in a loss of the movement through the Kelvin cycle. Okay? And so this loss in the movement through the Kelvin cycle means that the plant has a reduced ability to make sugar, a reduced ability to actually continue the process of photosynthesis. And so when do we see this? Well, we see this most when it's hot out, okay, And we're going to come back to that highlight in a second here. But when it's hot out, because when it's hot, if the stomata are open, then water will be lost. Okay, now water is not directly related to the Kelvin cycle, but the plant itself needs to keep that water inside. So when it's hot out, the plants will close the stomata. Okay. When the stomata is closed, that's where we see a reduction in carbon dioxide, because the carbon dioxide that's in there is going to be used up through the process of carbon fixation, and we see a high oxygen concentration. Well, this high oxygen, or the increase in oxygen concentration, is related to the high light. So in sunny areas is where we see plants that have this issue. In the desert, areas where there's a lot of sun. And this high light means that there's a lot of light energy to be captured by the light reaction, which means a lot of water will be split, releasing a lot of oxygen. And so plants that rely on the Kelvin cycle alone have this issue, is that rather than going through carbon fixation, they're going to go through the process known as photorespiration. And photorespiration is the process by which rubisco begins to break down the ribulose bisphosphate. So that's bad for the plant, because the plant's going to lose the ability to continue the process of photosynthesis, in particular to continue the dark reactions. And so now we need to talk about the processes by which plants have evolved in hot, arid, hot, dry climates to overcome that. And so most plants that only use the Kelvin cycle, and therefore only use rubisco for carbon fixation, are the ones that are going to have a problem. And these are known as C3 plants. Okay? So the only pathway in C3 plants is the use of rubisco to fix carbon. Okay? They're referred to as C3 plants simply because they have this 3-carbon intermediate that's formed at the beginning of the Kelvin cycle. So these plants grow well in our area, not a lot of sunlight, not too hot most of the year. In areas where there's a hot, dry climate, think of Arizona, the Southwest, okay, other desert regions. In these cases, the stomata remain closed for a longer period of time. So if we took a C3 plant and we took it out to that, stomata is closed for a longer time, which means oxygen is building up inside the plant, which is reducing the photosynthetic efficiency simply because rubisco is beginning to go through the process of photorespiration. So evolution steps in at this point and says, we need a way to overcome this. And evolution, plants have evolved 2 different mechanisms by which they've overcome this. Okay? And these are known as the C4 pathway and the CAM pathway. Okay? The bottom one on this one is the CAM pathway. And so I'm going to describe it this way so that you can see what happens. C4 pathway uses a spatial, okay, or space separation. It separates processes in the leaves into 2 different locations. Okay? CAM plants, on the other hand, are going to use temporal separation. In other words, they're going to separate based on time. Well, some of you are sitting there listening to this and saying, but what are we separating? Okay. Well, the answer to what we're separating is we're separating the process of carbon fixation from the process of the Kelvin cycle. Okay. So these plants we'll start with the C4 plants because they both use the same alternative pathway. But the C4 is giving you the name. If C3 plants are using a 3-carbon intermediate, your automatic thought should be these plants are using a 4-carbon intermediate for carbon fixation. And that's what we're seeing here, okay? And so this is the C4 pathway demonstrated here, and it's going to use an enzyme known as PEP-C, okay, or phosphoenolpyruvate carboxylase. And the phosphoenolpyruvate carboxylase is going to attach the carbon dioxide to a 3-carbon intermediate, phosphoenolpyruvate, to form a 4-carbon intermediate, oxidyl acetate. So there, hence the name C4 pathway. The C4 pathway will then be able to take the carbon that was picked up through the carbon fixation here and move it into the area where Kelvin cycle is and release the carbon dioxide to then be used by Rubisco. Okay? So Rubisco is still present in these plants, but in this case it's separated either by time, as we'll talk about in a minute, or by location to be able to allow photosynthesis to occur without being disrupted by closing the stomata. Okay. And the way it works for this, for the C4 plants, the C4 pathway, or PEP-C, is in the outer cells, and so in this area, O2 can be low. Okay, O2 can be high, and CO2 can be low, but that doesn't affect PEP-C. PEP-C is a higher affinity for carbon dioxide, so it continues to fix carbon even when carbon dioxide is at a low concentration. And so this pathway is placed in the mesophyll cells, and these cells are near the surface of the plant, near the opening of the leaves. As we can see in this particular image, where we're looking at the mesophyll cells, which are these outer cells here, okay, and the mesophyll cells are going to be exposed to the area outside where carbon dioxide is low and oxygen is high. But since they're using PEP-C rather than using Rubisco to fix the carbon, they're not affected by that. Once the carbon is fixed by the C4 pathway, it's then moved into the bundle sheath cells. And if you'll notice in this picture again, the bundle sheath cells are more central, they're protected, the oxygen concentration can remain low because we're separating it into a different location, and so there's no longer this problem with oxygen being high and Rubisco being interfered with or going through photorespiration. Instead, we can maintain the low oxygen concentration because there's a separation in location. So that's the C4 plants. The C4 plants use this separation where carbon fixation is in one type of cell where oxygen is high, carbon dioxide would be lower, and using the C4, the PEP-C enzyme, whereas Rubisco remains deeper in the cell where we can use that carbon dioxide that's been fixed to maintain high carbon dioxide concentrations in that location and reduce the amount of oxygen exposure. And that is the separation that the C4 plants are using. It's almost like we have two different rooms. They have an outer room here, the mesophyll cell, where carbon fixation is occurring, and an inner room here where carbon dioxide can remain high for Rubisco and Kelvin cycle to occur. So that's the first type of evolutionary change that has occurred. The second one are CAM plants. Okay? And so CAM plants have evolved a different mechanism in that they separate based on time. So CAM stands for crassoclean acid metabolism. And no, I don't worry about that name. It's just simply put is what's happening is at night when the stomata can be open bringing in the carbon dioxide, it's going to be fixed using the C4 pathway. It's then going to be stored as organic acids. And these acids, okay, that's where the CAM comes in. These acids will maintain the carbon dioxide in there or store it so that then during the day when the light's out and there is a lot of sunlight that's able to produce the ATP that's needed and the NADP that's needed, okay, they can then release their carbon dioxide to maintain high carbon dioxide inside the leaves and allow Rubisco to continue to function normally. So this is a temporal separation. At night, carbon dioxide is fixed, forming these organic acids that are stored until the day when the sunlight's out, and the sunlight in this case is needed for the light reactions to form ATP and NADPH, that then can allow carbon dioxide to be maintained high to keep Kelvin cycle going. So these are the difference, or this is the difference in the two types of pathways that have evolved to allow plants to overcome the limitation of Rubisco. So one last note that I want to make here, and that is the question that maybe some of you are asking now, why don't all plants just use a C4 pathway or a CAM pathway? Well, the answer to that question is energy. Okay? And you'll notice here, this is where we're getting the energy. For the C4 pathway to function, it requires excess ATP. And so in areas where there's a lot of sunlight, that's not a problem. Okay? So in areas that are hot, arid, a lot of sunlight, the plants just simply are able to continue to make ATP. In areas where there's not as much sunlight, western Pennsylvania, for example, the plants want to conserve as much energy as possible, and so they avoid the extra energy requirement and simply use Rubisco or the C3 pathway. So I hope you can see in this the difference in the two pathways and the advantages or disadvantages in each location. Again, this is all necessary because when oxygen is high, carbon dioxide is low, C3 plants or Rubisco will begin to go through photorespiration rather than carbon fixation. Okay. Thank you for watching again. And as always, if you have questions, please post them on the discussion board on Schoology or contact me and I will try to help clarify that. Thank you.